Welcome everyone to this ACEMS virtual public lecture, Playing God with Virtual Hearts. Before proceeding, I wish to acknowledge the people of the Kulin Nations on whose land we're gathered today at Monash, and I pay my respects to their elders past and present. Please note that this lecture will be recorded and made available on the ACEMS website, which is www.acems.org.au. And for those of you unfamiliar with ACEMS, ACEMS is the Australian Research Council Centre of Excellence for Mathematical and Statistical Frontiers. My name is Tim Garoni. I'm a Chief Investigator with ACEMS and the Deputy Director responsible for outreach. And I'm delighted to welcome Dr. Brodie Lawson to present today. Brodie obtained his PhD in applied mathematics, creating computer simulations of cell proliferation and migration. The majority of Brodie's postdoc career has been associated with ACEMS one way or another. And his research brings together ideas from computational mathematics, Bayesian statistics, computer graphics, and geometry in order to better understand the problem of signaling dysfunction in the heart. He now works as a research fellow with QUT's Centre for Data Science and as an associate investigator with ACEMS, collaborating with researchers from around the world, including Oxford, Brazil, and the Czech Republic. Brodie's research into creating virtual populations of heart cells that accurately represent experimental data, done in collaboration with other ACEMS investigators, including Chief Investigator Kevin Burridge, has been published in the prestigious interdisciplinary journal Science Advances. So at the end of this presentation, we will be taking questions. Please use the Q&A button on the bottom of your screen to post questions. And Brodie, welcome. And thank you for joining us today.
Yeah, thanks a lot for the intro. And I'd just like to acknowledge the traditional owners of the land here at QUT as well before getting started. I'll just start my timer. And let's jump into it. Okay, yeah, thanks a lot. So I wanted to start with an introduction to the heart because there's a lot more to it than you might initially think. And for people who have seen my talks before, a lot of this may be familiar to you, but I think it's really interesting, so I like talking about it. And so I want people actually just to think, for 3 seconds, because time, do you know what actually makes the heart beat? So, have a silent little think to yourself. And maybe, this is what I always used to believe. You think, oh, well, the brain probably controlling it, right? And, the brain's not consciously thinking about it, but subconsciously, it's making the heart beat the same way as it's probably telling us to breathe and everything else in the body. But that's not actually quite true. And so I've got a little statement there. Yeah, the brain doesn't really directly control it. And, you can make your comments about being in the wrong kinds of love there if you want. But what's actually going on is the heart is kind of self-exciting, self-regulating. And I say exciting because it's actually waves of excitation that cause it to beat. So you have this tiny little node sitting in there, and it is able to trigger itself to get excited. And then it spreads that excitation to the neighboring cells. And that actually causes a heartbeat through, a little explanation that we're about to go through. And so actually, the brain isn't directly controlling anything. That little node is just sitting there beating by itself at a certain rate. All the brain can do is control the rate. And even then, it's not really direct control. So here's just a little diagram of how the heart is kind of put together, especially from the electrical perspective. So these electrical waves of excitation are what makes it beat. And they start at that little node. They flow through the atria, which are the top chambers, which are the smaller ones. And they mostly just gather a little bit of blood and push it into the ventricles, which are the main ones at the end there. But then you go through another node to kind of protect the ventricles from anything funny going on in the atria. And you shoot through a lot of little fibers that carry excitation quickly so that the ventricles can all be activated kind of at the same time. And you get a nice synchronized thing where all of the cells see the signal to beat. And they do that, at roughly the same time or, in a coordinated fashion that gives you a blood pumping effect. And so the big thing to note here is you have that node protecting the ventricles from the atria if there's funny business going on in there. So that's why you have diseases like atrial fibrillation that are, diseases with negative effects, but you can live with atrial fibrillation and, potentially not feel it too much because the ventricles are protected. On the other hand, if you have fibrillation in the ventricles, you can have minutes to live, and that's why we have defibrillators around. But we'll see a bit more about that later on. Okay, so the individual cells that are actually doing the beating in the heart, what they do is they wait for a signal, which is calcium ions. They get a calcium signal, and then when they see that, that causes them to contract. And calcium does a lot of signaling in a lot of different cell types, actually, but that's the one that's triggering heart cells to contract. But for them to get that calcium from, the surrounding tissue, what they actually need to do is open their calcium channels, and they don't do that until they see excitement, and then they get excited themselves. And so you have this little graph here of the voltage, but they essentially sit at rest until they detect stimulus, either because you jolt them with electricity or their neighbors say, hey, I'm excited. And then they let a bunch of sodium ions in very rapidly, spit out a bunch of potassium, which causes the decrease back down. And in the middle phase here is when the calcium channels open, and you get this plateau where in and out currents balance somewhat. And it's in that phase that you get the signaling. Okay, and so we will see some more of these little graphs. You don't really need to understand all of the fine details there to understand it, but the point is that cells, they beat when they see calcium, and to get the calcium in, they need to open up other channels, which means they need to get excited. And that little plot just kind of shows their response over time. Okay, and the other thing that's nice about those plots, you probably, or it depends how many of my talks you've been to, I guess, you might not have seen action potentials before, which is that graph on the left, but you're probably familiar with ECGs. And so ECGs are actually just averages of the action potential, like the voltage values across the whole heart. So, you put your probes on different sides of the heart, different parts of the body, and then you detect the voltage difference across those, which is done by, an integral over your heart. And this is what the dynamics of the actual individual cells are inside that integral. Okay, so in one way, heart cells are very smart. They have a lot of different ion channels that govern that intake and outflux of ions, the potassium, the calcium, and the sodium. And they do this in a way that responds to what I've called here polarization state. But that's just, the voltage, how excited is the cell at the moment? Like, what is the difference inside the cell and outside in terms of how many ions it's got? And so by controlling in response to that, it does all those things that I talked about and a whole lot more. So, like, oh, I'm not going to open my calcium channels until I've detected that I'm excited, because that means I'm supposed to beat. Or, yeah, I'm only going to spit out a bunch of potassium when I need to repolarize myself back down to rest so I can be ready to beat again later. And part of the reason we have that, that big plateau and all that time taken to get back to rest is so that we don't trigger the heart again too quickly. But, even with this funny, complicated picture, I'm not going to explain any more than that. This is actually still a very simplified representation. And you can go into a lot more detail because you can look at individual channels and the stochastic probabilities with which they open and close, which the next level up of models will average over, et cetera, et cetera. So this is actually still very, very simplified, even though it's complicated enough, and you'll see a bit more of it a bit later. On the other sense, though, heart cells are also very dumb. And that's because if the sodium channel's open, then they go and do their little graph. They go and do this little pattern of excitation here, because that's what they're programmed to do. And so that's what I was saying before. The brain's certainly not controlling them. The heart itself isn't controlling individual cells. They're all just sitting there waiting for a signal. And if they get the signal and they're in the resting state, then they will just go and excite. And they'll start, a new wave of excitation potentially. And they have no idea, an individual cell sitting there in the heart has no idea if it's a good idea to do that or not. I think it's the order of 10,000 or maybe 100,000 maybe cells. So, an individual one doesn't know what it's doing. And what that means is, when the sodium channels open through those conditions, you can get very bad things happening because the heart is on autopilot. And so we can see here an example. So this is the atria So it might look kind of shaped like an actual heart, but that's actually just the atria rotated in a sort of funny angle. But this is just showing traveling waves moving through the atria. And so those are just causing a synchronized beating of the atria. You don't see it beat because we're not simulating anything except the electrical part. But those are just causing contractions of the heart, and the atria would then be pushing that blood through the valves into the ventricles, ready to be pushed around the body by the contracting effect of the ventricles. So that's what happens when things are normal. But because heart cells aren't smart enough to know what they're doing individually, you can also create things like this. So here, this is just in 2D tissue, and we use some kind of artificial initial conditions to kick it off. But this is a very real thing that is happening in people with cardiac diseases, hearts, which is here, oh, I've got a spiral wave. And so you might have seen spiral waves depending on your mathematical background, but what this is really saying for the heart people is, that little node that's supposed to be sending out the waves that trigger the heartbeat isn't what's triggering the heartbeat. This little spiral is just happily sitting in there, living forever. It'll just keep spinning around until it runs into a boundary, or something else happens with the interaction of the other waves coming from that node, perhaps. So what that means is that little node has now lost control over that heart chamber. That spiral will just keep triggering it at its own rate, whatever the intrinsic rate is, which depends on all the parameters of the process, of which there are a lot. But the point is, yeah, we now have a very rapid heartbeat in this chamber, and it's not being controlled by the body directly anymore. It's actually just coming purely from this spiral. So that's bad. I mean, a heartbeat that's too fast and uncontrolled can unduly stress the heart, which then causes other damage, which can make things get worse. But the worse is this, at least electrically. What can go wrong electrically? So just by changing a couple of parameters in the model that we're using to simulate this, now we actually just get complete chaos. And so this is fibrillation. Previously what you saw there would have been called tachycardia, which is just a too fast heartbeat. This is fibrillation. So this is chaos. And so if you look at it again and you kind of imagine the boundaries of this, when are they getting activated? And you see them get hit by waves, but now you suddenly start seeing different parts of the boundary hit at different times. You've got complete chaos as to when different parts of the tissue get excited. And what that means is you lose coordinated pumping function. So if someone is having, like a sudden heart attack, what that often means is their ventricles, the part that actually does the pumping of the blood, is doing that. And that means, yeah, that's when you have minutes to live, because that's the issue where you don't actually have coordinated function. And so you're not really transferring enough blood, or maybe much at all. And that's why your ECG kind of goes, closer to a flat line, because you're averaging over noise and getting essentially zero. Okay, and so this is actually why defibrillation works. The whole reason that you jolt someone with electricity and it magically makes them, come back to life, as it's portrayed, is because if the heart has fallen into that pattern of, disastrous signaling that's, triggered by itself, the body's lost control over the heart, what we want to do is we want to reset it. And what's the way that we reset it? Well, we just trigger all the heart cells to get excited and then relax at the same time. Or that's what we're trying to do. And the way that we try to do that is we just jolt across the whole heart with a whole lot of electricity. And I haven't studied any deeper into defibrillation, so that might be a slightly butchered explanation, but that's the key idea behind it. So I find that really interesting to kind of understand, oh, so that's what's actually going on there. The other thing that I find interesting to realize, the little facts that I like to say when I talk about this heart stuff to people, firstly is that electrolytes, I heard about them all in the sports drinks, and it's like, oh yeah, we put all these electrolytes in and they, help you go. And it sounded kind of like, made up pseudoscience that a lot of these advertisers will use. But no, the electrolytes that they talk about are actually the sodium, potassium, and calcium ions. And they have a very real and direct effect on the heart. And certain types of malnutrition can cause some of the problems like you saw on the previous slides, because your heart cells are now, not in their normal parameter ranges anymore, because you've changed the balance of ions around them. Okay, so how do we compute a heart? There's a couple of questions to that, because this talk is about virtually looking at hearts. So we need to be able to compute them. First question, do we mathematically understand a heart? And the answer is pretty well, actually. So there's a lot of organs in the body that aren't as well understood. I would say the heart is probably mathematically best understood. A lot of work has gone into understanding the heart, which is bringing together mathematics and experimental data. Sometimes the way in which that happens is a bit clunky. And so what you see here on the screen is just a whole bunch of screenshots. Well, it's actually, yeah, cut up bits of one long screenshot, I guess, from a MATLAB implementation of one ionic model for a single heart cell. And you might look at that and be like, oh, cool code. You might look at this and say, well, those formulas look very silly. So the people who are, mathematically inclined could look at things like 1 divided by 1 plus the exponential of e to the negative v plus 26 on 7 and say, that doesn't sound like a mathematical description of something. And you'd be right. That's been curve fit to experimental data. So because of all those complicated processes that are happening under the hood, so things like the stochasticity and the ion channel expression, when they actually open and close randomly on an individual level, we're averaging out over a lot of that. We're averaging out over a lot of things that are going on, even to come down to this level. And so there is, a lot of sort of, very empirical fits in here. And some questions about the best way to do that are some of the big questions that actually remain when I talk about mathematically understanding the heart, because people do, can write down a mathematical model that simulates heart tissue pretty well. You saw some simulations of it already, because I like to put videos in, wherever I can get an excuse to. But people could argue about, well, whose model is the best, because there are competitors and they all have different approaches. They're all being parameterized differently. There's actually a lot of investigation still to have there. And the reason people can argue about that is because maybe there's not one right answer. We have this issue of variability, which is what a big part of the talk is going to be about. Okay, but so let's say, yes, we can mathematically understand a heart. Can we computationally simulate a heart? And that in itself is actually a big question. And so here I just have a slide that will just talk a little bit about, what are the issues with trying to do this? And so I've been talking about the electrophysiology side, the electrical signaling that makes the heart beat, because that's the stuff I know. That's actually just one part of the story. But even just looking at that one part and trying to do it properly is tough. You saw that we're talking with traveling wave systems. They're sharp-fronted traveling waves. And it might take 2 milliseconds for the cell to go from rest to excited, but it takes somewhere around 300, 400 milliseconds for it to go back to resting. So that means, and the heart might be beating at 60 beats per minute. So that's once every second. So we have excitation that takes place over the scale of milliseconds, but one single heartbeat is 1,000 of those. And so if we need to use very fine discretizations in time to actually, cutting up individual milliseconds into lots of small pieces, you can imagine then if you want to simulate multiple heartbeats, you're looking at trouble. And then if you extend that out to minutes or hours to investigate, dynamic effects that creep up on you over time or things like that, you're really looking at trouble. And that's without mentioning that because these because these traveling waves are sharp-fronted, we also have to discretize space really finely. So numerically, this is actually a challenge. But that's one part of the story. The next part is the actual mechanics. So there are models out there. Honestly, I haven't touched them that link the ionic flow that's actually controlling the electronic stuff to, oh, I've seen the calcium, so now I'm going to actually contract. And so there is actually, extra models that link that in, which only makes it more numerically complicated and time consuming. And then you also have blood flow, which I think mostly has stayed separate as a domain from what I've seen, but that's computational fluid dynamics. So that's also, not any guarantee to be able to simulate well or quickly. So yes, we can computationally simulate a heart, but it's quite challenging actually. And here's the next question is it worth it to simulate a heart? And so obviously, you know, the talk is, well, let's play with virtual hearts. So the answer that I'm going to say is yes, but there's some good reasons why, I think. And so one of the big ones is this issue of variability. Depending on your background, you might have heard it called intersubject variability, which is, you know, the variability between different subjects, for example, different people in a population. And so I went and grabbed this from a random reference, trying to look, you know, just as a broad example, sort of outside of specifically the heart. And so I've just highlighted the important text here that I thought, which is you have this measure of effect, the absolute response difference. And so antidepressants and placebo are only 17% different. And you think, oh, okay, so does that mean that antidepressants are a scam? And not really, because there'll be a lot of people who take antidepressants and then, you know, start feeling a lot better. There'll be a lot of other people that take them and don't feel much of an effect. And that happens for all kinds of drugs. The reason that, you know, the antidepressants are highlighted there is just because it was a very small percentage. It's a nice sentence to summarize it, but the whole paragraph is saying a similar thing, which is most drugs, they don't work consistently. And that's the big story. Because people are different, their bodies are different, their pharmacological needs are different, they might metabolize things differently, but then also, you know, the root cause of the problem might actually be different. There's a lot of variability from person to person, and I don't think we're really fully addressing it yet. And so that's why I'm calling it the next frontier, because even in a case like the heart, where we mathematically understand things very well, there's still a lot of variability. And if you're trying to get real experimental data, especially in people, you're looking at very small numbers of, you know, sample sizes that are very variable and trying to learn from that. And that's why I'm going to be arguing, you know, well, computation can help here. And so the second part highlighted there is just saying, yeah, out of 17 common pharmacological treatments, only 11 managed to actually average out to a minimally clinically important difference. So 6 common pharmacological treatments on average don't even do enough to be minimally effective. Again, because, you know, it's variable how people respond to them. In the heart specifically, we see these things as well. So one good example is dofetilide, which is a class 3 anti-arrhythmic agent. But what that really just means is you give it to someone, and it's like a drug, so it could be taken orally or however else. Don't ask me actually to quote specifically how it's done for dofetilide. But the point is, it changes that ionic signaling process in the hopes of terminating dysfunctional signaling patterns like you saw in the movies. And so we have this anti-arrhythmic agent, and one of its adverse effects is torsade de pointe, which is a type of arrhythmia. And so you sit there and you think, okay, this is an anti-arrhythmic agent, and one of its adverse effects is literally arrhythmia. And that might only happen in a, you know, 5% is kind of the average of that range. But 5% of cases getting arrhythmia from an antiarrhythmic drug is obviously not good. And I'm not even just kind of exaggerating this, because dofetilide has actually been removed as a common antiarrhythmic medication because it was being used to treat arrhythmias in the atria. And as I said before, the atria is kind of protected from the ventricles, which are the important ones. Torsade de pointe happens in the ventricles. And so it leads to things like, you know, risk of sudden cardiac death. And so dofetilide, there were trials done with it, and it actually essentially killed a whole lot of people, which is quite grim, but a really stark sort of highlighting of this importance of variability and understanding, you know, when can we give a certain drug or another? How do people respond differentially to it? Another example taken from a paper that I'll mention a bit more later too is, you know, we can take, in this case, it is a virtual population, as we'll discuss, and you can try an experiment on them and then find that they respond differently. And so this is representing variability across people, and a lot of people responded very normally, and then a few people, the heart cell suddenly starts, you know, self-oscillating or failing to repolarize altogether, or, you know, taking a very, very long time to repolarize. Obviously, it's lost its normal behavior. Bad, bad, bad. So we might like to know, you know, when does that actually happen? And so there's just another example here of 8 different ECGs. So this was actually, human data, ECG data, but just shows the variability in the ECG across 8 different people. And that variability is coming from a bunch of places. So you have differences in the electrophysiological stuff, different levels of ion channel expression, which can depend on things like, hormone levels, and then also age and genetics. If you've ever heard of things like long QT syndrome, it's like a specific genetic disorder, but what it does is it causes certain ion channels in the heart to be expressed differently, and that causes cells to take longer to repolarize, which can be bad, and you get a long QT in the ECG. You can also have structural variability, and so this one is very important in terms of things like obstacles. So I won't go into details about this because I've already hit people over the head with a lot of heart stuff, but fibrosis is just like dead tissue, essentially, scar tissue in the heart, and it blocks conduction. So it can mess up electrical signaling too. And we have variability in, where that's positioned. And then you can have differences in behavior over time of heart cells in even a given person. Okay, so a little highlighting here of just, how much variability there is. On the left is kind of just another graphic making the same point. But on the right, you can just look through those dot points and see what kind of variability you actually have. So individual cells vary, different regions of the heart vary, the cell types are all different. So, atrial cells versus ventricular cells. Person to person varies, as we've talked about. If you have stress, and I meant that as in heart stress, although I'm sure, cortisol from general emotional stress would have effects too, and disease states, that will also cause the heart cells to adapt. So for example, with the chronic atrial fibrillation, where your atria is just constantly fibrillating, all of the cells in your atria need to respond to those new conditions where they're getting beat really quickly so that they don't, get stressed out and fail completely. So all of their behavior then changes in response to that disease state. And then also over time, you'll see variability. But that's enough about that. This is probably a more interesting slide. This is just making the point about why structural variability is important. So this is some simulation here where we created kind of a scar region. All of the little dark blue dots are blocked regions, and then we send a wave of excitation through it. And both of them have just been created with a random realization, just some density in the middle that tapers off towards the outside. But just very different results will emerge from what is essentially exactly the same thing. And if you zoomed out and looked at that, you would think, oh yeah, that's the same thing. There's no difference between those two. But one of them creates a self-exciting structure, a substrate for an arrhythmia, and the other one just happily emits a wave and nothing happens. And so you can see, as it keeps going, it just keeps spitting out waves. And so again, that's another example of a self-exciting structure in the heart that's going to make it beat too fast and just keep happening over and over again. Okay. So how do we learn about variability? We can go out there and try and take, lots of experiments, and then at best, or, at least we could average over all of the different kind of variability that we see. And that is one approach, but we get a lot of questions when we try, because, if I take a heart, do I know anything about what ion channel expression it actually had for the cells in it? Not really. I could try and measure it, but I need to measure that individually because that's a very small scale measurement. If you measure one type of ion channel expression, you can't measure other ones at the same time. So that's kind of already not feasible. How many hearts can we draw from? Well, if we want human data especially, that is a limiting factor in itself. If we wanted to look at things like the effect of those blockages, fibrosis, do we know the patterning of fibrosis? And actually, sort of 2010s-ish, but we have started using MRI to detect fibrosis using some clever tricks biologically, but that is only able to do kind of the resolution of MRI, not the fine scale structures that you saw on the previous slide. Other things that we have to worry about, if I do a bunch of experiments in the lab, what was the temperature in the lab? It could be different to the body. And you might say, well, you could heat the lab to 37 degrees, which I guess, the poor experimenters. But what I've seen instead is people in papers making arguments about, and to correct for temperature effects, we do this. And then if you want to do invasive experiments like on the left, you know, especially then, how do we get access to, you know, this many hearts to do that? And that is, you know, a very big limiter in terms of sample size. And you also have biases like we can only do proper experiments on hearts when we have access to heart patients. So they tend to, you know, skew older, for example. So there's a lot of issues like that. But if we come into code world and we want to look at variability, what do? Well, I'll just change the temperature. Or, okay, I want to simulate some disease state. I'll just play with these parameters to represent disease state. So we're kind of able to peek under the hood here by doing it this way. And if we want to do it this way, we have to make sure that we can describe and realize reality. And essentially, those are the issues from before. You know, can we mathematically describe the heart? And so I've showed some code for one cell. Just wanted to super quickly show that what we're dealing with mathematically is reaction diffusion, PDEs, well, a PDE, and it's coupled to a whole lot of ODEs, a system of ODEs, which represent all of those ionic transfers going on in the cell. And then to actually simulate it, we need to do some fancy stuff. I'm not going to talk through things there. I'm not going to claim that it's amazingly fancy, but what you're looking at there is kind of a control volume, finite element discretization with a second order sort of exponential integrator. So we're using a lot of tricks to try and get as much performance as we can because of all those issues in just, you know, time scale, space scale. How do we, you know, simulate quickly enough? And so let's assume that we can do that. I mean, we can. It's just computationally demanding. What can we actually do? Well, if we can create these virtual populations where we can actually peek under the hood, you know, we can see what the numbers are in the code. We know all the details. And, you know, we're limited only by computing power. We can do any kind of experiment on them that we want, and we'll see some examples of that. And we can call back in anyone. So it's like, oh, your data turned out to be interesting. You know, what were your details? Well, we'll just go, look, we've got it saved. It's a, you know, it's a file on the computer. We can have that guarantee of grabbing anyone of interest. And we can actually use this to try and understand variability without understanding a full system, as we will see. Okay, so the big kind of landmark paper, and this is from the group at Oxford. I can't claim to be attached to this work, but what they did was they showed that they could run in silico drug trials, in silico meaning in silicon, as in virtually. It's kind of like, you know, the extension to in vivo, in vitro, that stuff, if you haven't encountered that language. But so what they did was they said, you know, let's do virtual drug trials on a bunch of virtual people and see how they respond to different drugs. You know, is it actually one of those risky ones like the fetolide that we talked about before or not? And they found that they had better predictive accuracy than the animal studies that people are usually doing for this. And so that's another thing. Animal hearts are quite different to human hearts. And we actually mathematically describe animal hearts with their own models. It's gone to that level. And that's because of, you know, access to data, essentially. But there is actually a lot of animal experimentation happening in the heart. And so there's organizations like the 3R, which aim for the replacement, reduction, and refinement of animal testing. And so this, you know, these in silico trials are actually kind of a big emergent from, you know, that grant. So if we want to create good virtual populations, though, that reflect data we have, we need to make sure we're matching the character of the data. And so what people have done in the past is just said, well, you know, I'll generate virtual souls at random. I've got lots of parameters. I know there's variability, so I'll just make them random. And then if it looks like a human, I'll accept it in. And it's literally just, do you fit in the box that looks like a human? Because what they actually did was drew a box around the limits of the data and said everything that falls in that box, you're golden. And if you can look at that, you know, demonstrative image there, you can see if you draw a box around the X's, which is the data, you can get a lot of red points that would be fitting into that box, but not fitting the data. And what we'd like to do is get the blue instead. Okay, so we're going to try and select the blue. And the way that we did this, which is the paper from Science Advances, we looked at it as a kind of Bayesian parameter calibration problem, but it doesn't quite function as a normal one of those. You have to go hierarchical to do it properly. So instead, we took the normal, you know, Bayes theorem, and we said, nah, we're just going to look at data density. So, you know, like how many people in the population on average seem to have values around here compared to over there. So we represented the data as a distribution. And then we said, well, let's accept and reject according to, you know, that density. We used sequential Monte Carlo. I'm not going to go into any kind of Bayesian sampling details because I know, you know, time will definitely be a factor. But with that, just quickly say, if you are interested in Bayesian parameter calibration, I do recommend that as a method. It can be actually quite user-friendly. Just don't look at the original papers that published it because they're very technical. Okay, but we had to correct because as I've put that little kind of blue highlighted bit there, we were kind of, you know, closing our eyes and saying, yeah, well, we'll go by density. We need to fix that. And so what we see here is a bunch of little biomarkers, which are measurements from that curve. The white is data. So here they had individual cells from about 300 people, which is a very good sample size for the heart. And they've got a spread of how those biomarkers varied across that population of 300. If you look at the white, you can see they're spread over a very big range of values. And so what we did was we tried the box approach, which is the red. And you can see some very boxy corners where you can see that exact effect I was talking about happening in practice. And then we tried our sequential Monte Carlo, which is the blue. And you can see it does well, but it has some issues still. One big one is down here where the model itself, you know, the variability and the model together can't actually predict the data. So you need to re-examine assumptions there. But if we use the refinement step and we ignore, you know, the one where we have issues, actually we're looking pretty good. So the light blue now is doing a very good fit to the data after we pick that kind of optimal subpopulation. And, you know, this is just showing the marginal distribution. So it's just another way of showing, yeah, it fits, except for the one that the model couldn't really deal with, which you can see doesn't fit. Okay, so what I'm arguing here is we've got now a more representative virtual population. And this virtual population, if you apply it to the data for healthy people and the data for atrial fibrillation people, you actually find that it calibrates nicely and grabs out what are the important differences in, you know, those cell response curves between the, you know, healthy people and the cells that have adapted to that atrial fibrillation condition. And if you use the box approach, you don't actually really get that. So it is actually really important to do this calibration well, but if we can get some very neat things out of it. The first thing that you see over here on the right, so this is showing the original base model with just the parameter values that were published when the model itself was released. You can see that it didn't really match the data in terms of like where the APD90, which is one of the biomarkers, you know, don't need to worry about the detail, but you can see it didn't really fit the data that well. But it does show the overall trend of, you know, a spiky one for the red and then a rapid jolt down and then a hump for the blue, which is what we got for our population, our virtual population. But what we can then do with our, you know, calibrated population is go and do some virtual experiments. And so what we actually see here is let's hit them with, in this case, dofetilide. I mean, we can do that now because we're not going to be harming anybody. Let's hit them with dofetilide and see how they respond. And the key effect, which is making the cells take longer to recover, so it's harder for a wave to come back around and re-excite them, we do get that effect. And we didn't get that in the original model that was, you know, not fitting our data, just had, you know, default parameter values. So we actually found by using the virtual population, we were able to recover what we would expect. But now we can also highlight, oh, what about the virtual members of our population we want to call back in to look at more? Well, we can pick out these, you know, aberrant behaviors and say, what's going on with you? And unlike someone that we could just, you know, in a real experiment, we could come in and say, you know, what's going on with you? And they'll say, oh yeah, my, you know, IKR channels are 90% as expressed as the rest of the population. No, they can't say that. But, you know, we've created that person, we've specified these parameters for them. So we actually know, you know, that member of the population, what's going on with them to the, you know, under the hood level of detail, which is what we need to investigate these things. Oops, sorry. So the next part I just wanted to talk about was the idea of mass experimentation. So previously I was talking about calibrating models where you have, you know, you got some data and you try and get some representative people. Now I'm talking about let's just create lots and lots and lots of virtual, in quotes, people and see how they respond across, that whole variability sort of range expressed by all those people. And so it, again, just comes down to computing power. And so to deal with that, we try and, all of these people, just in this case, crudely copied in Paint, we want to get all those people and we want to crudely experiment on them on a large scale to exhaustively understand what's going on in the heart. And so here's an example on the left of machine learning doing that. This is not a real person. It's from the website. This person does not exist. They've trained a machine learning algorithm to just create fake photos of people. And I'd seen some of the other ones, but the person one is actually really, really good. You can spot, oh, something went a bit wrong here. But I mean, you see the face first, and the face looks pretty much dead on. So yeah, that person doesn't actually exist. That's a surrogate person created by, a machine learning algorithm. And that's what we're actually doing too. I mean, ours looks a lot less dramatic or interesting. But in another sense, I think we've actually done pretty well there. So that was a complicated set of dynamics just showing how the character of spiral waves responded to different parameters in the model. And we've filled out the whole surface between all the red points and the white points, which is the training and test data. We've filled out that whole surface, and it's a very complicated surface, as you can see, but the machine learning goes and fills it out. And so now our surrogate people would be all the people who aren't on the red and the white spots. We can get all the other people from the population and see how they behave overall. One way we can do that, that's just saying that, it works well enough. I won't talk about that. But yeah, so how do we do that? Well, we can average over all of these virtual people that we've just kind of, they're very crudely modeled, but that's okay because we're just going to average over them. We just want to get an overall sense of how our model behaves. And so what we do is we do a Monte Carlo integration where we remove all of the parameters but one. Statistically, we'd call that marginalizing out one parameter. But we do it with a Monte Carlo integration where we just run the surrogate model, which is our crude model that came from machine learning. And that will then let us get a sort of average effect. They actually call these the main effects. And you see a lot of noise over here for some of the ones that, have a very complicated effect and or the method is not working precisely well for those. You see other ones that behave completely as we would expect. Don't have time to talk through them. But then you also see some interesting unexpected things. And that's what we want to learn. That's what the virtual population is coming along and telling us. Here, we found out that if you make cells take longer to recover their excitability, usually seen as a good thing because it makes it hard for a traveling wave to loop back around and hit the cell and find it ready to be excited again, and then, cause the spiral to actually continue. So normally you'd think, yeah, making them delay longer is good because they'll take longer to get excited. It's harder for a spiral to come back around and hit them. But what it actually also does when you delay that is you create lots of room for potential like partial activations, and that causes the chaotic patterns that then devolve into the fibrillation chaos. So we actually found that the organization, which is like a measure of chaos, goes down when you actually delay recovery. And so the drugs out there that delay that recovery, that's how they work. This might show, in the cases where they don't work or they have an adverse effect, why that's actually happening. This was another piece of work where we did some similar studies to identify what parameter regime was kind of the risky one in terms of spiral waves. Well, yeah, they get kicked off by the blockages, the fibrotic blockages. And here we were looking at the different disease conditions. So when the cells die and you create a scar region, it affects the cells that are still there living, but it affects them in different ways. So we were playing with what are the different ways that those cells get affected by like the change in pH, the extra potassium ions that are around, et cetera, and identifying what are the danger regions in terms of actually creating an arrhythmia. But we won't have time to talk more about that. Instead, in this kind of smattering of results that I wanted to finish up with, I've got this one here with the cheeky name, this fibrotic patterning doesn't exist, because just like this person doesn't exist, we went and created an algorithm that generates new realizations of patterning based on one that you feed into it. So there was a little bit of hand designing the generator, but the actual calibration of that generator, it's the same generator for all four. It goes and learns the parameters it needs to make that sort of different type of patterning. So we're actually able to create these. And this is important because if you have a virtual population and you want to stick in representations of fibrosis, and these are the real patterns of fibrosis you see from histology experiments, well, then that now allows us to check, what variability will we see across those different virtual members, because we can create lots of people with very slightly different, but still indicative of the overall trend, patterns of fibrosis. Other thing that we looked at is, well, if I want to simulate that, and I've got all these issues of scale, then when I want to actually zoom out, I need to look at, on the bigger scale with those fine details captured. So we did some work with homogenization, where you try and replace all the fine scale details about what's blocked and what's not with just like a big block of representative material. In this case, you could do, oh, well, it's kind of red and white, so it averages out to pink. You could say, well, hang on, but I have more vertical flow than horizontal because I got these big blocks that would, I'd have to work around them to go horizontal. So I might try and learn a bit more about what character the flow there has. You can see those up arrows there. And, there's a bunch of maths kind of behind that, but I won't go into the maths for it. Instead, I'll just show kind of the philosophy in effect. So there you have a scar region being stimulated by spiral waves. And you can see that it actually stabilizes the spiral wave. So it doesn't degenerate into chaos because of the scar region, which you might say, good, I'm not getting the fibrillation, I'm not getting, minutes to live. On the other hand, you might say bad, because now I know this is going to persist forever. It's not going to randomly annihilate itself because it's chaos. And we just show here, if we average out into, big blocks of representative material, you can see the big squares there. And the visualizing effect looks a bit funny. And I mean, if you want to ask about it at the end, you can, I guess. But you can see that we capture the same effect of the spiral wave being stabilized, and also its frequency of rotation is also well estimated. Okay, so that's kind of one of the critical parts in simulating these methods when we want to generate all these fine scale patterns, but use them in actual regions of tissue that are big enough to have spiral waves. Oops, sorry. Okay, just quickly here, we also looked at better ways to calibrate, or maybe at least faster ways to calibrate. So the science advances where we said, here's a way to statistically calibrate. We have come back to that more recently, and this is something that's, in preparation. But we're looking at using a linear model to actually approximate the true model, and then doing some neat tricks to actually use that to calibrate a population very rapidly. And so you can see there an initial condition for a funny looking toy model. And then the final state that it gets to is actually what you see there. So you can actually see, the 3D view and then the 2D slice views. We actually have, blue dots and red dots that look like they're, both just samples from the same population. So we have been able to calibrate it even just using these crude linear models, which is a big time-saving technique because we haven't done all the Bayesian sampling. Okay, and that's about 45 minutes. So thanks for that. I hope you found the heart stuff interesting and kind of a glimpse into all the different ways that we can look at virtually then working on these heart models to learn all sorts of things that we just can't see looking at real people. I've said this is a thanks also for people tuning in, but I just wanted to also thank all the people that have been associated with this work, especially Kevin Burridge, who was my supervisor and kind of the lead on the heart project that a lot of this work has sort of come through. And I'd just like to also say, rest in peace, Edmund Crampon, who was studying the heart. And I had a good conversation with him when I visited Uni Melbourne about this stuff. And I was really, devastated to hear that. So rest in peace. Thanks for everything. Sorry for the sort of somber ending note there, and happy to take questions. Thanks.
Okay, thanks, Brodie. It was a very interesting talk. Okay, so there is one question in the Q&A. So the question is, if these infinite dysfunctional loops in your movies can explain conditions like, try and get this right, postural tachycardia syndrome. Do you have any thoughts on that?
Okay, so in terms of my technical knowledge, I couldn't tell you what specifically postural tachycardia is, but that's absolutely right that this is a type of tachycardia. So the normal heartbeat is, 60 beats per minute, for example, if you have you know, a healthy resting heart rate, you know, that's probably optimal, I guess. But those spirals can be rotating kind of 4 or 5 times a second in some of the more extreme cases. And actually, if I go back to here, we were looking at the frequency of rotation, the dominant frequency. So here, yeah, so 1 hertz is a healthy heartbeat. And those spirals that we were getting in that simulations were up at around, you know, 6 to 8 on average across the, you know, whole population and the sensitivity analysis kind of thing that we did. So that's indicating, yes, like a very, very serious type of tachycardia, because your heart is beating way more rapidly than it should be.
So if anybody else has any questions, please put them in the Q&A. But so I've got a broad one. So you said right at the start that the heart's better understood than any other organ. Is that just because it's more well studied, or is it somehow simpler?
I think it's because it's more well studied. I wouldn't want to make any sort of bold claims about the complexity of other organs, but I do feel like the heart is definitely up there, because you've got a lot of interacting pieces, but the interactions are kind of very, you know, clear and meaningful. It's like, so for example, all the electrical signaling is happening, and then that links in with the mechanics that actually cause it to pump. And then you have the actual blood flow part, which is, you know, fluid dynamics. So there's a lot of pieces there. So I wouldn't say that it's kind of the easiest, but it is very well understood. Part of that is because they were able to do a lot of experiments on similar cells. So you have the squid giant axon, which is what was studied by Hodgkin and Huxley in 1950 something, I think, or 1960. And so that was kind of, you know, Nobel Prize winning work that went into what is actually happening that creates these signals. And because it's the giant axon from like a big squid, you can actually see like the pulses through it as it contracts. So they were able to do things like jolt electricity and see, you know, self-exciting patterns and things like that. And that kind of then built into a mathematical model that tried to represent what was happening under the hood in terms of the ion channels for these cells. So I think part of it is they've had access to some nice experiments they could do. And there was kind of a nice history there that has then built into what is actually quite a big field. And I'll just quickly say, one of the challenges that I've found with this topic area is, because it is a big field, it is reasonably mathematically understood, and the simulations are very tough. One of the, like, you know, numerically, or, you know, setting them up, those kind of things, what that actually creates a challenge where you might have done some interesting new mathematics and applied it in a new way to learn some things about the systems, but people will come back and say, well, have you fully simulated a realistic 3D heart? No. Well, you know, some other people are for the work that they do, and the work might be different, but there's actually kind of that, you know, level of, you know, the state of the art, which is out there, and a lot of people are doing it. So it does create a kind of extra challenge there, I think, as well, because it's so well understood. And yeah.
So there's one more question in the Q&A. Can you talk a little bit about how you did the finite element modeling for dealing with the spatial variation of dynamics?
Yeah, so as I said, like, I don't want to claim that I've done anything super fancy, but I will say that you do have some things that are probably not heart specific, but probably specific to sharp-fronted traveling waves. These are sharp-fronted traveling waves that are not coming from an advection term. So we have, it's just reaction diffusion. It's just the reaction term, you know, can do very funny things very quickly. So what that means is you don't have dramas with, oh, my numerical scheme is introducing too much diffusion, I'm not getting sharp-fronted waves. Instead, you naturally get sharp-fronted waves if you go to a fine enough scale to see them, like you should. But you have all these issues with, if I want to diffusively cause sharp-fronted waves, I have this very sensitive cell waiting there to see enough excitation to trigger. And what that means is, for example, if you put cells too far apart, and like, so you're like, your grid spacing in your finite element or finite volume is too far apart, what will happen is it will kind of diffuse out before it ever gets above that threshold. So if you actually extend your spatial scale too big, you just actually see no propagation of waves at all. And if you go, not too big, but bigger than it should be, you'll find that the traveling waves move a lot slower than they should, because you're not really getting that oomph of extra pulse from the next cell until it's diffused out to meet it. So if it's too far away, it really slows things down. And so it creates this effect where these types of models, when you simulate them, they're very, very sensitive to the spatial discretization, more so than the temporal one, even though the temporal one has, very, very fast dynamics happening in it. And you have to be careful with things like, for example, mass lumping, which is very common in finite element methods. But if you do it here, then you only ramp up that effect of, not having a very good spatial integration and, your wave moving too slow unless you go to a really, really, really fine grid. And so the method that we've used, we were looking at control volume finite element methods, which was essentially you're still doing the finite element thing of integrating over an interpolant and not lumping. So you're using, like we're using linear or bilinear, but then doing that in a control volume setup so that you have the properties of like conservation. Hopefully that answers your question.
Okay. I think we're pretty much out of time. So let me thank you again very much, Brodie. That was an excellent talk.
Thank you.